Hello, we are Ryan and Ishita, and our presentation is on the 1986 Nobel Prize in Physics, Scanning Tunneling Microscopy. The 1986 Nobel Prize in Physics was split between several people. One half of the prize was awarded to Ernst Ruska for his work on electron optics and for designing the electron microscope. The second half of the prize was split between Gerd Binnig and Heinrich Rohrer, 1/4 each for their design of the scanning tunneling microscope. Before we can cover scanning tunneling microscopy, it is useful to understand the progression of microscope technology over time to understand why scanning tunneling microscopy was such an important development. Optics have been around for a very long time. The earliest lens people have ever discovered dates to before 700 BC and is essentially just a chunk of quartz that was roughly ground down to a lens shape in order to magnify text. Around the 14th century and onwards in Europe, people began to use glasses to correct vision, and lenses became much more common. The center picture is a painting by Tommaso de Modena in 1352 of the Dominican cardinal Hugh of Saint-Cher, showing him reading with glasses. In the early 1600s, scientists began to build compound microscopes with multiple lenses. The microscope pictured on the right was made by Giuseppe Campani from around 1660. It was a copy of Galileo's microscope from around 1624, which Galileo had built after seeing one by Cornelius Drebbel exhibited in Rome in 1622 and thinking that he could improve on its design. As microscopes got better and more popular, people used them to study various small structures. Robert Hooke famously used a microscope that he designed to look at cork and discovered the cell. Pictured on the left here is a page of drawings by Leeuwenhoek in 1680 of the anatomy of a flea, which he studied under microscopes. Leeuwenhoek was one of the first scientists to study biological samples, and he publicized his findings. Lastly, around 1850, microscopes became commercially available and were rapidly adopted for various studies of small structures. Although optical microscopes were revolutionary for their ability to study microbes and small-scale structures, there's a fundamental limit to the size of structure that they can resolve. First discussed by Ernst Abbe in 1873, the diffraction limit of a microscope is equal to the wavelength of the light that the sample is being illuminated by, divided by 2 times the numerical aperture of the lens, which is equal to the refractive index of the lens times the sine of 1/2 of the angle of focused light. Modern optics can have a numerical aperture of up to about 1.5. Assuming that you have a visible violet light of around 400 nanometers, you would maximally be able to resolve structures of around 133 nanometers. Obviously, there are lots of structures that are smaller than this, and this estimate is essentially the best you can get with visible light. So something better than optical microscopes were necessary to visualize smaller structures. Fortunately, if you leave visible light, you can get wavelengths much smaller. In fact, Louis de Broglie, in his 1924 PhD thesis, presented a relation between momentum and wavelength, establishing that anything with momentum also has a wavelength. Much of the groundwork for the electron microscope was laid out by the cathode ray tube. In 1883, Heinrich Hertz demonstrated that cathode rays could be deflected by a magnetic field. Working off of this, Hans Busch published in 1926 that magnetic fields could theoretically be used to create an electron lens, focusing a narrow beam of electrons to a point. Max Knoll then led a team with Ernst Ruska, which worked on electron optics. This group read Busch's paper and developed the electron lenses required to focus electrons on a sample. In 1931, Knoll and Ruska used this setup to image aluminum mesh grids on a screen below the actual electron gun. In 1933, they refined this setup, producing the first images that were higher resolution than an optical microscope. This type of electron microscope is called a transmission electron microscope and consists of an electron gun, lenses to focus the beam, the sample, and then further electron lenses to focus the electrons onto a screen or camera. For the development of this technique, Ruska was awarded half of the 1986 Nobel Prize in Physics. Unfortunately, by the time of the awarding, Knoll and several other scientists involved in the development had passed away. As the electron microscope improved, smaller and smaller structures could start to be measured. In the pursuit of even finer detail, 2 IBM scientists, Gerd Binnig and Heinrich Rohrer, began working on what would become the scanning tunneling microscope. The project started under Rohrer in the late 1970s, and Binnig was hired in 1978 to work on it. By 1983, Bittig and Rohrer had gotten the STM working. Here's a clip from an IBM video showing the STM that they designed.
At first glance, this apparatus itself doesn't look like a microscope. Its heart is inside the vacuum bell jar and consists essentially of a tip and a sample, a piezoelectric tripod to move the tip three-dimensionally with extreme precision, and a specially developed step motor to shift and position the sample precisely. An absolute essential for measuring is an almost completely vibration-free suspension, which also had to be specially developed. The measurement data are read and processed by a computer and displayed on a screen or plotter.
Very soon after the development, in 1986, Bittig and Rohrer were awarded the Nobel Prize. Modern STM setups are essentially the same as what they showed in the clip. Essentially, you have the sample that you are measuring, which is on a plate that can move very finely using piezoelectric stepper motors. You also have the actual probe tip, which measures the voltage between the tip and the sample. This is all processed by the computer, which provides feedback to move the tip and the sample. All of this is under vacuum on a vibrationally isolated setup. So all of that progression is how we ended up with the STM. Now Ishita will explain fundamentally how a scanning tunneling microscope works.
So picking up from what Ryan said, let's dive deep into the main principle of STM, that is quantum tunneling. As we know in classical physics, if an electron does not have enough energy to overcome the barrier, it should not be able to cross it. But in quantum mechanics, the electrons behave like waves and also as particles. And because of that, the wave function does not end suddenly at the barrier. Instead, it extends into the barrier and can even reach the other side. That is the reason the tunneling can happen, and so if the electron does not have enough energy to go over the barrier, there is still a finite probability that it can pass through it, and that is what we call quantum tunneling. In this image, the left side represents the surface, the right side represents the tip, and the blue region in the middle is the potential barrier. The red curve shows the electron wave function, and it is quite evident from this image that the electron is passing from one area to the other, overcoming the potential barrier based on these conditions. And this equation right here shows that the tunneling probability decreases very strongly when the barrier becomes higher or wider. So the most important point here is that the tunneling is extremely sensitive to distance, and that is exactly why STM can detect very tiny changes at the atomic scale. Now let's see how this happens in the STM. Here the tunneling process is shown in real STM situation. In the figure A, you can see that the sample and tip are separated by a very small vacuum gap, and also the direction of the tunneling is from the sample to the tip. Whereas in figure B, the direction is reversed and it travels from tip to the sample. And so the direction of the tunneling depends on the applied voltage. And that is important because the STM signal is not only controlled by the distance between the tip and the sample, it also measures the electronic process between the sample and the tip, and thus gives useful information about the surface structure and also about the electronic behavior at the nanoscale. Now, once we have understood how the tunneling current is produced, the next question is how we control the tip position with such high precision. And to answer that, STM relies on piezoelectric effect. The piezoelectric effect is what allows the STM tip to move in an extremely precise and controlled way. In the image, we can see that a 6-step actuation process of a piezo inchworm motor. This motion happens step by step in a very controlled manner. And in STM, piezoelectric scanners are used to move the tip in an X, Y, and Z directions. And this means the tip can scan across the surface line by line, and it can also move up and down to keep the correct distance from the sample. The key point is that this movement can be controlled with Armstrong level precision, and that level of precision is essential because tunneling changes drastically even the tip moves a very tiny amount. So without piezoelectric control, STM would not be able to maintain stable tunneling conditions or generate accurate atomic scale images. Now that we have looked in everything, let us see how the mechanism of STM works. In this figure, the sharp tip is attached to the piezoelectric tube with the electrodes and the sample is placed just below it. A tunneling voltage is applied between the tip and the sample, and when the tip comes close enough to the surface, the electrons across the gap and crosses the gap, and this produces the tunneling current. This tunneling current is measured using the tunneling current amplifier and then sent to the distance control scanning unit, and then finally processed and displayed as an image. Here in this image you can see the interaction between the tip and the atoms taking at the surface of the sample. And now let's know what are the factors on which the tunneling current is dependent on. The first one will be the applied base, and then the second one is the local density of states of the sample and also the distance between the tip and the spacing. And among this, the most critical point for imaging is the current changes exponentially at a distance, exponentially with distance. And because of that, STM can detect extremely small changes in the surface height and convert them into atomic resolution images. So, after seeing the basic mechanism, the next step is to see how the STM operates. There are two main operating modes in STM. The first one being the constant current mode, and in this mode the tunneling current is kept constant while the tip scans over the surface. And to do that, the tip moves up and down and depending upon the surface features, and that vertical movement is used to reconstruct the topography of the surface. On the right we have the constant height mode, and in that case the height of the tip stays fixed and the tunneling current changes as different surface features move under the tip. So here the current variation itself contains the surface information. The constant current mode is usually the more common one because it is safer and more suitable for the rough or uneven surfaces. Now let us see some applications of STM. The first one would be the atomic scale surface imaging. The images here you can see that the STM directly visualizes the structures on the surface at extremely small length scale. In the image A, we can see a clear periodic pattern which reflects the atomic arrangement on the surface. And in images B, C and D, we see that it compares different STM images contrast with structural models and helping us connect the observed features with actual atomic or molecular arrangements. In image E, we can see a large area scan that shows an ordered pattern across the surface, while in F, we can see that it highlights the local features and the possible defects. Next, moving on to the next example, here in this image, this is the chemical structure of the molecule that is being studied, and here is the schematic diagram of the STM tip above the molecule which being placed on the gold surface, and this explains the experimental setup where the tunneling current is measured. And in this figure it is seen that the current as an function of time at two different voltages, which gives the information about the changes in the molecular state during the measurement. And lastly, from D to H shows a sequence of molecular images with the color scale representing the signal intensity from low to high. As the shape changes from one image to the next, it suggests that the STM can follow changes in molecular electronic state or molecular configuration. So, this example shows us that STM is useful not only for the imaging atoms but also for probing single molecules and studying their behavior at nanoscale. Now, moving on from the examples and the capabilities of the STM, I would like to finish with a few concluding points and some future directions for it. So to conclude, STM is a very powerful technique for studying the surfaces and materials at the atomic scale. Its operation is based on the quantum tunneling together with high precision tip control through the piezoelectric effect. Now, let us discuss some of the future directions. And the first one would be integration of STM with AFM, which combines the atomic imaging with the force-based information, and some more complete surface characterization can be seen from it. And the next one would be STS, which measures the local electronic state by recording the tunneling current at a function of voltage. And the last point is the development of a next generation integrated scanning probe method where STM is combined with time resolved and multifunctional techniques to study ultra fast nanoscale processes. So, overall, STM has developed from a surface imaging method into a much broader and more advanced tool for nanoscale science. Thank you all for your attention. Thank you.